Hello everyone, welcome back to our channel Pathshala, your one-stop solution for all the subjects related to dairy science and dairy technology. So in today's video, we are going to discuss about differences between prokaryotes and eukaryotes. To give a little context of this video, I would like to tell you that we are running a lecture series on the subjects dairy microbiology, and first we are going to cover fundamentals of microbiology. So in the last video, we have discussed about the history of microbiology, in which we have discussed something about cell, who coined the term cell, and I want to raise a question to all of you that who coined the term cell and in which year? I hope you know the answer if you have watched the video. Otherwise, you can click on the link given below and you can answer in the comments. In today's lecture, we will discuss about the key differences between prokaryotes and eukaryotes, and before that, we will also see what is the term prokaryotes and eukaryotes mean. So the things which I am going to cover in this video is the introduction about the types of the cells which are present, key similarities between the two types of cells, and then we'll go to the main part of our video, that is the key differences between the two types of the cells, and then we'll discuss some important applications, how these things will help in our future dairy microbiology. And at last, we will summarize the whole lecture. So, firstly, we'll see what prokaryotes and eukaryotes are, or the type of cells prokaryotic and eukaryotic are. So, as we know, we have discussed about cell. So, cell have two types, or in our world, they come in two basic forms, which are known as prokaryotic and eukaryotic. So, as we can see, that this karyos word comes from a Greek language word, which is known as literal meaning kernel. So the students from agriculture background may know this term kernel. It is a part of a grain, right? So in terms of microbiology, we use this word to refer to the nucleus of a cell. Now at this stage, I want to tell you a simple trick or a technique which you can use. If you don't know the meaning of a word, which is a difficult term like prokaryotic or eukaryotic, and since the word is not native to the English language, what you can do is you can bifurcate the word, and after bifurcating you can have two different words from the same word, and then you can find the meaning of those two words, and if you join the word those two words you can find the whole meaning. By doing this way you can remember that word for your lifetime because you have put such an effort to find the meaning of that word. So we'll go with the same technique. For example, let's take prokaryotic. So prokaryotic is made by joining two words, which is pro and karyos. So pro meaning before and karyos meaning cell or a nucleus of the cell. So what does it mean? So pro means before a nucleus or a cell which was existing before a proper nucleus is developed. And in similar way, if we talk about eukaryotic, so it is also made from two words which is eu and karyos. So again, eu meaning true or good and karyos meaning a nucleus of a cell. So what does eukaryotic mean? Yes, it means a cell which possesses a true nucleus. So this defines the basic difference between the two types of the cell, prokaryotic and eukaryotic, that one possesses true nucleus, which is called as eukaryotic, and one doesn't have true nucleus, or a cell which exists before a true nucleus, it is called as prokaryotic. So again, the same thing, prokaryotic cell have no nuclei, while eukaryotic cell do have true nuclei. So here you can observe a 3D diagram of a prokaryotic cell. So you can see that the different parts of the cells are not properly placed at their locations. We can say that they are not well mannerly placed in their proper locations. So this is how a prokaryotic cell looks like. An eukaryotic cell, so you can observe that all the different parts of the cells, this is the nucleus and these are the DNA and the ribosomes, are all placed at their particular place. So which we can, in the definition we have already seen that it is called as a eukaryotic cell which have a well-defined nucleus. So before moving to the main topic of our video, that the key differences in the prokaryotes and eukaryotes, first we will see and find out about some key similarities between prokaryotes and eukaryotes. Because basically at the end both are a type of cells, right? So they have some key similarities in them. which they share amongst themselves. So both have DNA as their genetic material. So we know by definition that both of them are cells. So they need to replicate, right? They need to produce more cells to grow themselves. And for replication purpose, DNA is required. So it is very well understood that they both require DNA and they both possess DNA as their genetic material. Second thing is they both are covered by a cell membrane. So both have cell membrane. It is a type of a covering layer or a protective layer or inside which all the different parts of the cell are placed. So the next thing is both contains RNA. So RNA is nothing but ribonucleic acid. So it is used to create protein for the development, produce DNA for the growth and development of the cell. So both of the cells need their growth and development, and for it they require protein. So both of them contains RNA, and both are made from same basic chemicals. And what are these chemicals? The carbohydrates, the proteins, nucleic acids, minerals, fats, and vitamins. So all the basic nutrients and chemicals which are required by everyone like you and me. Cells also require the same chemicals for their growth and development. So both possess same type of chemicals. Now, both have ribosomes. So these are the structures on which proteins are made. As I told you, both contains RNA, which help in the creation of proteins. So ribosomes are the structures on which proteins are made. Since the protein is being made, so obviously both will contain ribosomes. I know these terms can be difficult for some of you, but these are biological terms. You need to memorize it or by heart it. But if you understand the function and if you can connect each one of it, it will be easy for you guys to remember the things. For example, as I've told you, RNA are used to develop and creation of protein. And how these proteins are made, by help of which structure these proteins are made, it is ribosomes. So if a cell is containing RNA, it is obvious that the cell will also contain ribosomes, right? So again, both regulate the flow of material and the waste that enters and leave them. For example, if you are a person and if you eat something as nutrients and after digestion something goes waste, and which will you extract, right? That the waste you will excrete daily on daily basis. Similarly, the cells also, they require nutrients for their energy and different mechanical processes or the metabolic processes. And some of the nutrients they utilizes and other goes waste. So they also have they regulate the flow of these nutrients, which whatever enters, it also goes out as excretion material. And both have similar basic metabolism processes, which are also known as life processes. What are these processes? For example, photosynthesis or reproduction, because cells also require food, they also need energy. How they can get energy? If it is a plant cell, they do photosynthesis, they convert carbon dioxide and release oxygen to convert it into energy. Or they do reproduction, which is a basic life process. They will replicate themselves to produce new cells. So both of the cells, either it is prokaryotic or eukaryotic, both perform similar basic metabolism functions, and both require a supply of energy. If they are performing these functions, they are regulating the flow of nutrients in and out of them, they are performing the metabolic processes. For performing all these functions, it is very natural that they require a supply of energy, right? If you wake up in the morning, you are going on jogging, then you are coming, getting ready for college, studying whole day, coming back to the college, going on the playground, playing, doing your homework. So these all things requires energy, right? That's why you are eating breakfast, you are eating lunch, and at the night you are eating dinner. So for performing various functions, your body needs energy. In a similar manner, cell also performs different functions. So to perform these functions, they require a supply of energy, right? All good. So now we will move to the next part. Both are highly regulated by elaborate sensing systems, which in common words we can call them as chemical noses, right? So cell doesn't have noses like us, like humans, so we can sense what is happening around us. But they require to know what is happening around them. Why? Because we have previously discussed. Just connect the previous points so you can easily remember it. In the last 2 points we have discussed that they both regulate flow of the nutrients. But how they know that in their surroundings this particular chemical is nutrient for them and this particular chemical is not a nutrient for them, it can be poisonous for them? It is done by sensing systems which are known as chemical noses. So by releasing some chemicals or by sensing some chemicals, they make themselves aware about the reactions within them and the environment around them, and then they can decide what to do with it, right? So these are known as chemical noses. These are similarly like our noses. Now we will move to the main part of our video, that is the key differences between prokaryotes and eukaryotes. So here we will see the basis of differences, and then we will see on how they are differ on that basis. So first basis is the nucleus, whether the cell possesses a nucleus or not. If yes, what is the type of that nucleus? So as we already read in the basic definition, that prokaryotic cells don't have a true nucleus, even though they have nucleus, but it is not true to itself. Not true to itself means that it is not well defined or it is not well structured, right? Their DNA floats in the nucleus. If it have true nucleus, DNA is embedded in the nucleus, right? So in eukaryotic cells, they have a distinct nucleus, like different nucleus, a proper nucleus which will perform the functions of a nucleus, right? So now the second part is that the main difference is occurs in the DNA structure. So prokaryotic DNA is circular and it doesn't have histones. So a student from the microbiology background, or they have a little idea about histones. So histones are nothing but type of proteins which find in DNA structure. This helps in the packaging of DNA material, because we know DNA holds our genetic information. So to package that material, we require histones. So prokaryotes, it doesn't have histones and the DNA shape is circular. Why circular? Because in the last point we have discussed that DNA continues to flow in the nucleus. It is not properly placed at one place, right? So that is why it has circular DNA. It keeps on flowing here and there inside the cell. While in the eukaryotic DNA, it is a linear DNA and it is associated with proteins which are histones, which we have just now discussed, right? So DNA, if anyone, if you know how it looks like a ladder, and both ladders are connected with different types of proteins which are called as histones. So if you have general diagram of DNA in your mind, it is normally a linear structure which we always read, but in the prokaryotic it is circular. Next point of difference in between both of the cells is the size and the complexity of the cell structure, right? So obviously eukaryotic cells contains true nucleus. They have a much larger body. They are bigger in size when compared to prokaryotes, and they have many organelles. So what is the term organelle mean? From the starting of the video, when I was showing you the diagram of the eukaryotic and prokaryotic cells, I used the word parts of the cell, different parts of the cells. So these parts of the cells, these are known as organelles. Suppose you are a cell, and in your body you have different organs to perform different functions, right? Similarly, if cell is a human being, so to perform the different functions inside a human being, as human being requires organs, similarly a cell requires different parts. These parts are known as organelles. So since eukaryotes have a well defined structure, they have various organelles to perform different functions. Each organelle have its distinct role. But in prokaryotic cells, they are very smaller and they have no membrane bound nucleus, or they don't have membrane bound organelles. What does it mean? That when in the case of eukaryotes, organelles have properly defined function, like each organelle will play its own role. Like, but in prokaryotic cell, it is not like that. For example, in our body, in human body, liver have distinct role, kidney have distinct role, heart have distinct role. But suppose if you are a prokaryotic cell, all the roles of these three organs will be done by a single organelle. Why? Because they don't have membrane bound organelles, they don't have distinct organelles to perform various functions, because they are smaller in size, they are less complex. They don't have well defined nucleus which can give work to different nucleus, right? So nucleus, we are discussing this term again and again. Nucleus is nothing but in a human body what work our brain plays. It is a main part of our body, right? It assigns the work to different organs of our body. In a similar manner, the nucleus in a cell, along with replication and holding the genetic information in form of DNA, it also assigns work to the different organelles. So since the prokaryotes doesn't have a well-defined nucleus, it is unable to define different roles for the organelles. So that is why they have less number of organelles, and one organelle will perform two or three functions. So they don't have distinct role and they are not membrane bound. Membrane bound in the sense they are not well defined. They are placed here and there, and they are not secured by a cell membrane, right? So then we will move to the next point of differentiation, that is ribosomes. So prokaryotic cells have simpler ribosomes, while eukaryotic cells have more complex ribosomes necessary for their larger structure and more functions. You need to connect all the points, right? In the similarities, what we discussed is both of the cells contains ribosomes to produce proteins, right? But since the prokaryotic cells are very simple in structure, they have less size and less complex structure, so they require less amount of protein or less amount of complex protein to be created. That is the reason they have simpler ribosomes. It is making sense, right? But for the eukaryotic cells, they have more complex ribosomes because it is required for their larger structure and to perform various functions. So it is obvious that require this type of cells. So now why we are discussing all these things here in dairy microbiology? We should discuss about different things, right? But why we are discussing how one cell differs from the other? What are the key similarities between the two cells? Because if you can look to this tiny diagram, this bacteria, this is known as bacteria, this is also a type of prokaryotic cell. This plays a very important and key role in our dairy industry. Many of us know this is called as curd or dahi, and this is the lassi, which we love to drink in the summer season, especially in the northern parts of the country. So these are called as fermented dairy products, right? And fermentation is a process by which these dairy products are made. So what is this fermentation process? This fermentation process is a conversion of milk sugar into acids to produce fermented products. And this fermentation process is carried out by these tiny organisms known as bacteria. And they, what type of cell these bacteria process? These are the prokaryotic cells, right? Along with the good microorganisms, we also have some bad microbes also. So we need to know their structure also so that we can design the processes in such a way that from good microorganism we can utilize them to their most of the potential, and from the bad microorganisms we can design the processes so that we can avoid the spoilage of the product, we can avoid the contamination of the product. Since ours is a food industry, it is of utmost important. So at the end, we'll about to finish our video. Just a quick recap whatever we have discussed in this video, that prokaryotic cells are simpler in terms of complexity. They are very smaller when compared to prokaryotic cells in terms of size. And when we go to the basic definition, they lack a true nucleus. They don't have a true nucleus. When it comes to eukaryotic cells, just go opposite to the points discussed earlier. They are larger in size, they are more complex in nature, and they have membrane bound nucleus. They have a true nucleus, and both the types of cells are essential to understanding dairy processes, especially the fermentation process, which we will discuss in the upcoming videos, and to understand the role of microorganisms in our dairy industry. So that's it from my side in this video. We'll see in the next video. Till then, don't forget to like, subscribe, and share the video if you like it, and also comment if you have any queries in the comment section. We'll try to answer all of your doubts. Till then, goodbye.